I'd like to welcome everyone to this module on plant cell culture. The assessment will be by a 1-hour open book exam. Because I have to run these exams during lecture slots, you need some time to arrive and get set up, then you need some time to do the exam, and we have to be finished by 10 to the hour. So I'm going to run it in 2 separate 1-hour lecture slots. It'll be in week 7, and in each of these you'll be given one experimental protocol in plant cell culture. So it may be micropropagation, or it may be plant nuclear transformation, or it may be plant plastid. You should minutely dissect these and make sense out of them, and you'll get 1 mark for every valid point that you make, and there will be well in excess of 100 points that you can actually make. But I would only expect somebody who is very good at plant cell culture to get near 100%, though it will be possible. So the types of points. In the protocol there's a sentence here: seeds were washed overnight under a running tap, rinsed for 10 seconds in 70% ethanol, and then sterilised in 20% domestos plus 0.1% volume for volume tween 20 for 10 minutes, followed by 3 rinses in sterile distilled water. This is typical of some of the information which may be provided in your experimental protocol. So this protocol asks specific questions. So why were they using seeds in the first place? So from your understanding of plant cell culture, you will have found out why seeds are a particularly good source of starting material for things like micropropagation or for plant genetic transformation. Why were they washed overnight? So there are specific reasons why seeds might be washed overnight. Why do it under a running tap? Why not just put them in a beaker? Why were they rinsed for 10 seconds in 70% ethanol? Why were they then sterilised? Why not use seeds that's not sterilised? Why use 20% domestos as a sterilant? Why add 0.1% volume for volume tween 20? So why use tween 20? And why use 0.1% volume for volume? Why not 1%? And why do it for 10 minutes? Why not do it for an hour or 2 minutes? Followed by 3 rinses in sterile distilled water. So why 3 rinses? Why not 1 rinse? Why not 6 rinses? Why use sterile water? And why use distilled water? Why not use tap water? So all these different elements of the protocol you will need to discuss and state the reasons why the particular protocol used these particular parameters. There are textbooks in the library. I will provide you with a lot of resources under Moodle, so there will be virtual lectures, there will be experimental protocols that you can go through, and there will also be some reviews on different techniques. So for the lecture outline, there's going to be the basics of plant tissue culture and micropropagation, plant nuclear transformation, and finally plastid transformation. And if you go to the experimental protocols, you'll find that these are broken down into a number of sections. So there are a series of virtual lectures covering the sections, followed by detailed discussion of a protocol. So just to finish up this virtual lecture with a grand overview of plant cell culture, your normal starting point will be some kind of explant. And this explant will typically need to be sterilized to give you a sterile explant. And this sterile explant can be used in a number of different ways: for micropropagation, for plant nuclear transformation, or something I'm not going to discuss today, which is product formation.